Organelles by Sneha Katuku and Sindhu Deveji.
Lesson overview. This presentation will teach you about the different organelles found in a human cell and how they function together to perform many biological processes. Questions you should be able to answer at the end of this lesson are: what are the different types of organelles and what are their functions and structures?
The mitochondria. The function of the mitochondria is to convert glucose, which is another form of sugar, into ATP, which is energy used by the body for many processes, and this happens through cellular respiration. The structure of the mitochondria is a double membrane, including the cristae, and this happens through endosymbiosis, where it forms the double membrane, as it originally started off as the prokaryote and was engulfed by another bigger eukaryotic cell. You may have also heard of the mitochondria as the powerhouse of the cell.
The nucleus. The function of the nucleus is to hold DNA, which is basically the information of the cell and the genetic code for the formation of proteins. Its structure involves a barrier known as a nuclear envelope, and it contains pores which allows materials to transfer in and out of the center. It is known as a control center of the cell since it holds DNA, which, as I mentioned earlier, is the information of the cell.
Chloroplast. The function of the chloroplast is to make food for the cell through the process of photosynthesis, which is taking light energy and converting it into chemical energy. The structure of the chloroplast is a double membrane structure that shows that it's most likely an organism engulfed by another organism. This is known as endosymbiosis.
The cytoplasm. The cytoplasm is a semi-fluid structure, meaning it's jelly-like substance, and it contains many chemicals to keep the cell functioning. It holds organelles in their place as well as other substances.
The cell membrane. The function of the cell membrane is it's a semi-permeable barrier that only allows certain molecules to go through, such as small nonpolar substances or materials, to enter and exit the cell. The structure of the cell membrane is a phospholipid bilayer, which means it consists of phosphate heads facing the aqueous or water environment and the interior lipids facing each other. So if water molecules, as they're polar, if they can't enter the cell membrane, there are many proteins embedded or on the top of the cell membrane, which allow the water molecules or other polar molecules to enter into the cell. This is what makes it a fluid mosaic model.
The lysosome. The lysosomes are spheres made up of a lipid bilayer that surrounds the hydrolytic enzymes contained within. Their main function is to break down different waste materials into non-toxic substances with the help of these hydrolytic enzymes, also known as digestive enzymes.
Peroxisome. The function of the peroxisome is to break down fatty acids used in creating the phospholipid part of the membrane and in cellular respiration. It also breaks down hydrogen peroxide, which is very toxic, that is formed from the oxidation reactions, such as creating the fatty acids, and it turns it into water, which is not toxic. The structure of the peroxisome is it's a single membrane-bound organelle with enzymes created by lipids and proteins found in the cytoplasm.
The Golgi Apparatus. The Golgi Apparatus is primarily composed of flattened membrane sacs. It has an entrance and exit known as a cis and transphase, respectively. Its main function is to collect synthesized molecules from the rough endoplasmic reticulum, package it, and ship it to its desired location, often through the process of exocytosis, where it leaves the cell.
Vacuole. The function of the vacuole in plant cells is to help with structural support as well as maintaining osmotic balance, which is the balance of water in and outside the cell. It also helps with waste disposal, storage, as in different materials and substances, and protection. The function in animals or all cells is to hold solutions that are created and stored or are engulfed by the cell. It keeps the cytosol from being exposed to the contents inside. The structure of the vacuole is a chamber surrounded by a semi-permeable membrane.
The smooth endoplasmic reticulum versus the rough endoplasmic reticulum. The rough endoplasmic reticulum It contains ribosomes on its surface, making it rough. These ribosomes are then used for synthesizing proteins, which are then shipped to the Golgi apparatus. The rough ER is primarily composed of flattened membrane sacs. However, on the other hand, the smooth ER are basically tube-like structures that synthesize lipids and detoxify harmful substances. It does not contain any ribosomes on the surface, making it smooth.
Listen to this fun rap song and other videos about organelles. Here are 3 links from Amoeba Sisters, Crash Course, and Bozeman Science that will help you understand organelles better.